At the end of the year 2013, we learned something really astonishing about the human genome. It contains not only one genetic code, but two. And this second code seems to explain an old observation about how genes are translated into proteins. As you all know, genes are transcribed to RNA, which is spliced, and then the processed transcript is translated into proteins in the ribosomes. The process of translation uses the genetic code by which groups of 3 nucleotides, called codons, are recognized by specific molecules, the transfer RNAs, that carry specific amino acid. But the genetic code is degenerate, meaning that the same amino acid can be encoded by 2 or more codons. In theory, codon usage should be uniform. That is, if 2 codons code for one amino acid, then cells could use either of them with equal preference. However, this is not the case. In most cases, there is one codon out of all synonymous codons that is used preferentially, a situation known as codon bias. And interestingly, not all the species show the same type of bias. So if one codon is used preferentially, for instance, in mammals to code for alanine, a different codon might be used in amoebae or whatever. Why such bias in codon usage exists is a matter of debate, but now, at least for the human genome, we have a new piece of information that gives us a more complete picture of what is happening. Scientists were analyzing the human genome in great detail, searching for regions to which transcriptional factors bind. As you remember, these binding sites are regulatory elements which switch genes on and off precisely through the binding of a specific transcription factor to a sequence motif. You need a certain sequence of a few nucleotides for the transcription factor to bind efficiently and then regulate the expression of a gene in the vicinity. Textbooks say that the vast majority of these regulatory elements, these short sequence motifs, are located in the non-coding part of the genome outside the exons. But surprisingly, what scientists found was that around 15% of all the codons in the human exome, the exome is the coding part of the genome, the collection of all exons put together, well, 15% of the codons are in fact occupied by transcription factors in some type of cell in the body. So this subset of codons seems to encode 2 different types of information, which is why they have been named Duons. On the one hand, they are interpreted by the genetic code to make proteins, that is their classical function. But they also contain a different code about which we had no idea at all, that is interpreted by the transcription factor machinery to regulate gene expression. And it is precisely this second hidden code, the one that seems to impose an evolutionary constraint because, as I say, you need a very specific sequence motif if the transcription factor is to bind efficiently at that point. So that sequence must be maintained during evolution in order to keep it functional. It cannot change. It seems that this constraint is, at least in part, what has led to the bias in codon usage that we have known for decades. And it might also explain why codon usage is different in different species.